We've talked considerably about protein structure and its relation to proper protein function. I suppose a good way to close our discussion of protein structure is to look at examples of problems that arise from improper protein folding. Let's first remind ourselves that the internal environment of the cell is very crowded. There are many proteins present. Given the number of proteins generated, it's not surprising that proteins occasionally fold incorrectly. Recall that the hydrophobic effect is what drives protein conformations, folding hydrophobic residues on the interior. If a protein misfolds, it often leads to extensive hydrophobic regions that are water exposed. In this case, the hydrophobic effect drives the spontaneous aggregation of these proteins. Since other proteins are in such close proximity, it may also mean that a misfolded protein can induce a misfold in another protein. Molecular chaperones are proteins whose function is to aid in the correct and timely folding of proteins. A case in point is the heat shock protein HSP70, also called DNA-K. This was actually the first chaperone discovered in a bacterial cell. Chaperones are present in all kinds of organisms, though, from simple to complex, emphasizing their vital role in living cells. All chaperones that we know of recognize a misfolded protein due to the exposed areas of hydrophobic residues. There are several diseases that result from the aggregation of misfolded proteins. Examples include the human diseases Alzheimer's and Parkinson's, and scabies in sheep. The picture below shows a dark patch in the brain indicating the formation of a plaque of aggregated proteins. These diseases all have as their causative agent a misfolded protein known as a prion. The properly folded form is known as PRP for prion protein and is pictured in the center of the slide. Notice that the secondary structure is 100% alpha helical in nature. The conformation changes in the misfolded form, known as PRP-SC, and is pictured on the far right. The letters SC stand for scrapie. Notice that the secondary structure now contains regions of beta sheets. With such a regular array of strands, perhaps you can see how easily these might aggregate into the structure illustrated on the far left. These are but practical examples of the extreme importance of proper protein folding.